Hello doctors. Welcome to Note Official. Let's break down medicine into concepts. Today we are going to explain embryology week 2. In the previous video we covered week 1. As we know, by day 7 the trophoblast has differentiated into cytotrophoblast and syncytiotrophoblast, and we have the inner cell mass. We will now see the further process in this video. Let me tell you one thing before we move forward, that the trophoblast will become the structure that provides nutrition and blood to the embryo. The inner cell mass will form the embryo itself. Implantation begins at day 6 and invasion starts. And now on day 8 invasion progresses, and the embryo invades endometrial cells further. A cytotrophoblast surrounds the inner cell mass. Syncytiotrophoblast continues to invade deeper into the endometrium. The inner cell mass is also known as the embryoblast because it will eventually form the embryo. The embryoblast now differentiates into two types of cells. Hypoblast will form extraembryonic tissues. These are cuboidal cells. Epiblast will form the embryo itself. These are columnar cells. Together, the hypoblast and epiblast form the bilaminar embryonic disc. A cavity forms between the cytotrophoblast and epiblast. This is called the amniotic cavity. Now what do we have on day 8 is embryoblast differentiates into epiblast and hypoblast. Bilaminar disc forms. Embryo invasion continues. Amniotic cavity forms. And now on day 9 syncytiotrophoblast invades further into the endometrial cells. The opening is now sealed by fibrin, also known as the coagulum, fibrin plug. This ensures the embryo is fully embedded. The hypoblast forms a membrane called Heuser's membrane, which creates the primary yolk sac cavity. More than half of the embryo is now implanted inside the endometrial lining. So we have coagulum fibrin plug formed. Hypoblast forms Heuser's membrane primary yolk sac cavity. Amnioblasts cover the amniotic cavity on day 9. Now what do we have developed until now? We have amniotic cavity, primary yolk sac, and more than half of the embryo embedded in the endometrium. Now as we move further to day 11 and 12, embryo is completely implanted inside the endometrium. We now have two cavities, yolk sac cavity and amniotic cavity. A cytotrophoblast grows toward the syncytiotrophoblast and begins to form the placenta. A loose connective tissue develops between the embryo and the syncytiotrophoblast. This will form the extraembryonic mesoderm. Syncytiotrophoblast invades the endometrial capillaries and begins supplying blood to the placenta. This is known as uteroplacental circulation, which begins at day 12. Villi-like structures appear that supply blood to the placenta. These are called primary chorionic villi. Primary chorionic villi contain only syncytiotrophoblast and cytotrophoblast at this stage. On day 12 embryo implantation is complete. Early uteroplacental circulation begins. Loose connective tissue forms. And primary chorionic villi present. As day passes, cytotrophoblast continues to grow further towards syncytiotrophoblast. The space between the loose connective tissue and the trophoblast forms the extraembryonic mesoderm. From the yolk sac, a duct-like structure grows toward the placenta. This is called the Allantois. The yolk sac is now referred to as the secondary yolk sac. So now we know that hypoblasts form primary yolk sac and it grows to secondary yolk sac. Since we now have extraembryonic mesoderm. In addition to the primary chorionic villi, the villi upgrade to secondary chorionic villi. Secondary chorionic villi contain syncytiotrophoblast, cytotrophoblast and extraembryonic mesoderm. On day 13 to 15. Now let's understand extraembryonic mesoderm. So extraembryonic mesoderm has two layers. One layer is only surrounds the yolk sac, which is known as splanchnopleuric layer. And second layer is surrounds the entire embryo, known as somatopleuric layer. This is amnion formed by amnioblasts and somatopleuric extraembryonic mesoderm. And this is chorion formed by trophoblast and extraembryonic mesoderm. Now at the end of the week 2, we have two types of trophoblast, syncytiotrophoblast and cytotrophoblast. Two types of embryoblast, hypoblast and epiblast. 2 cavities, amniotic cavity and yolk sac cavity, and 2 layers of extraembryonic mesoderm splanchnopluric layer and somatopluric layer. That's why week 2 is called week of 2. Let's revise week 2 briefly. On day 8, trophoblasts differentiate into syncytio-trophoblast and cytotrophoblast. An invasion continues. Embryoblasts differentiate into hypoblast and epiblast and form bilaminar disc. Amniotic cavity forms between cytotrophoblast and epiblast. Now on day 9, a coagulum forms, and it makes sure that embryo is fully embedded. A hypoblast forms Heuser's membrane, which will make primary yolk sac cavity. Amnioblasts cover the amniotic cavity. Now day 11 and 12. Implantation complete. A loose connective tissue forms extraembryonic mesoderm. Uteroplacental circulation begins with primary chorionic villi. Day 13 to 15. A extraembryonic mesoderm with 2-layer splanchnopluric layer and somatopluric layers. A secondary yolk sac and Allantois form. Amnion and Chorion form. A primary chorionic villi grows into secondary chorionic villi. We will see the further process in the next video. Make sure you subscribe the channel. Now we solve the question before ending the video. Which of the following is not a part of secondary chorionic villi? Option A. Extraembryonic mesoderm. Option B. Syncytio-trophoblast. Option C. Cytotrophoblast. Option D. Splanchnopluric mesoderm. Next question. When does uteroplacental circulation begin in the embryo? Option A, day 5 to 7. Option B, day 7 to 9. Option C, day 11 to 12. Option D, day 15 to 17. If you want to solve more quizzes, follow us on Instagram, @NoteNoteOfficial. Thank you for watching. See you in the next video.